Hello, everyone, and welcome to my conference talk at today's symposium. My name is Sneha Upadhyayula. I am 17 years old and I am from Hudson, Ohio. Today, I will be presenting a conference talk on Epstein-Barr virus as a disease-causing agent in well-characterized disease states and the broader population. I will also be looking at the predictive applications of this research in developing screening and preventative tools. I want to begin my presentation by sharing a quote by Napoleon Hill, which I feel captures the true essence of any important advancements we make in science, research, and the broader world. Knowledge has no value except that which can be gained from its application towards some worthy end. In our collective societal pursuit of a greater knowledge base, there have been far too many instances in which valuable information that has already been uncovered is rendered ineffective by the failure to apply it where it can make a difference. In choosing my topic for this research project, I was inclined toward exploring Epstein-Barr virus because it is a field where there is so much available information, yet so little at the same time. This struck me as a clear issue because it limits the application of the existing knowledge base to clinical efforts in treatment and prevention. In my presentation, I aim to address common instances of EBV-induced oncogenesis with a focus on highlighting the pathways that EBV takes to make this possible and ultimately drawing underlying conclusions about how this information is relevant in the development of new preventative and treatment procedures. And just as a side note, anytime I use the abbreviation EBV, this is in reference to Epstein-Barr virus. Here's a table of contents that outlines the path of my presentation today. I will begin with a core question, then move to an introduction, followed by the methodology used to obtain sources for this review, displaying the results, providing my interpretations of these results, and finally coming to a conclusion where I will discuss how we can use this research in practical application to improve treatment and preventative methods. Let us begin with the core question. What factors and states cause a usually benign Epstein-Barr virus to turn a host cell into a tumor lineage? And how does understanding these factors contribute to the development of new clinical screening procedures and treatment methods? This is the thesis question to keep in mind as we move throughout the different parts of this presentation. I want to begin by going over some key terms that I will be using throughout this presentation. First, Epstein-Barr virus, or EBV, our primary point of discussion today, is one of 9 known human herpes viruses in the herpes family. It affects 90% of adults and is the most common cause of infectious mononucleosis, a syndrome with fever, sore throat, and large lymph nodes and fatigue. It is most commonly transferred through saliva, but after infection, the virus remains in latent condition in host B lymphocytes. Oncogenesis refers to the process of cancer formation, specifically when normal cells are transformed into cancerous cells. An oncogene refers to any gene that has undergone mutations such that it has the potential to cause cancer. B lymphocytes are one type of lymphocyte or white blood cell in the immune system. Also known as B cells, they're responsible for creating antibodies, which can then bind to pathogens or foreign substances to attack them or neutralize their impact. Epstein-Barr virus latently resides in the B lymphocytes, however, when reactivated, can be responsible for the development of many types of B cell cancers. Natural killer cells, or NK cells, are another type of lymphocyte responsible for destroying any cells that are infected, damaged, or diseased. And finally, cytotoxicity refers to the characteristic of being toxic to a cell. Cytotoxic substances often cause harm to cells and can even increase cancer risk. However, cytotoxic drugs have been reverse engineered for use in cancer treatment, such that the drugs are toxic to the cancer cells themselves and thus limiting their ability to proliferate. Now, to briefly introduce the subject and provide some background, Epstein-Barr virus, or EBV, affects most adults worldwide, but typically resides in a latent form in the body's lymphocytes. Though this virus is consistently found to latently reside specifically in the peripheral blood memory B lymphocytes and some bone marrow cells, in rare cases, natural error via cell malformation and repeated proliferation can allow infected cells to spread and cause cancer. A better understanding of the virus and its effects in normal populations is necessary and valuable in helping with immunization and protection for high-risk groups. I also want to quickly outline the methodology here. This literature review primarily obtains its sources from the PubMed Central Database. All searches outlined a scope from database inception to April of 2023, and some keywords used to conduct the searches were Epstein-Barr virus, oncogenesis, nasopharyngeal carcinoma, X-linked lymphoproliferative disease, post-transplant lymphoproliferative disorder, classic Hodgkin lymphoma, and serological markers. To reiterate what I described earlier, Epstein-Barr virus infection affects 90% of adults and is the most common cause of infectious mononucleosis. It is commonly transferred through saliva, but after infection, the virus remains in latent condition in host B lymphocytes. It is generally kept in check by the host's immune mechanisms. However, in rare cases, EBV can reactivate or replicate unfettered and lead to multitudinous reactions within the body. Let us begin by discussing some of these paths that EBV takes in both oncogenesis and infection. Epstein-Barr virus has been directly linked to a variety of different cancers, transitioning infected cells to a cancerous form due to abnormal gene expression. Generally speaking, EBV DNA binds to the surface of host DNA in a normal cell and rewires the molecule such that it completely changes its shape. This change in shape has been linked to the activation of many cancer-related genes in the body. However, EBV may also take other paths in viral oncogenesis. By binding to the host DNA, EBV can cause chemical changes to the epigenome that result in the switching off of tumor suppressor genes. Without the control of normal immune mechanisms, cellular apoptosis cannot be activated as the cell is not aware of the incorrect integration that has happened. Apoptosis refers to cell death, and this is the function that cannot occur because of this incorrect integration. The malformation of a single cell, followed by monoclonal proliferation of that damaged cell, results in many of the malignant tumors that we know to be associated with Epstein-Barr virus. This lack of immune response is better understood in the context of the EBV oncogene EBNA1. The EBNA1 oncogene is responsible for maintaining the genome of EBV in latently infected cells. However, EBNA1 can also inhibit NK, or natural killer cell responses, and apoptosis reactions to cells that were recently infected. EBNA1 also causes there to be significantly less surveillance from the immune system, thus allowing EBV to result in carcinogenesis without being stopped by the body's natural defense mechanisms. EBV also contains many proteins, such as the EBNA3C antigen, which allows for the transformation, proliferation, and survival of the infected lymphocytes. These proteins suppress the transcription process of necessary parts of the cell cycle, and thus the cell lacks essential parts for its function, even if it were a normal cell. These cells should be destroyed immediately, and yet apoptosis reactions cannot be fueled because the virus has shut off all appropriate immune reactions. Even the pre-apoptotic tumor suppressor genes and factors, such as promotion of the G1 or S phase, are suppressed by these oncogenic proteins. These proteins essentially inhibit each part of the cell cycle so that damaged cells can continue to proliferate and result in many of the malignancies that we are aware of. All the pathways mentioned above can occur in any patient. However, specific medical conditions are known to render patients especially vulnerable to these mechanisms. One such example is X-linked lymphoproliferative syndrome, or XLP, a genetic immune deficiency. In this condition, the affected group primarily being young males, a mutation within the SH2D1A gene affects the receptor involved in T cell and natural killer cell signal transduction. This is a silent immune deficiency. However, when a person with XLP is met with primary EBV infection, they could die from fulminant infectious mononucleosis syndrome or EBV-induced lymphoma because of their limited immune function. About 50 to 60% of people with XLP can develop EBV-related complications, which include malignant lymphoma in about 30% of these patients. There are no simple methods for genetic screening. However, XLP can be found by gene sequencing in the case of the previously mentioned conditions. This screening is critical because if the T cells and NK cells become dysfunctional and fail to control the unfettered replication of EBV-infected cells, cell signals, and receptors that are sending incorrect cells, this will result in the failure of apoptosis to occur when necessary, ultimately resulting in malignant tumors. Another example of one of these well-characterized disease states is in the case of post-transplant lymphoproliferative disorder, or PTLD, a condition in which groups of B cells grow out of control after a patient has received immune suppression for an organ transplant. EBV-associated PTLD can occur in up to 20% of solid organ transplant recipients, with the risk depending on a variety of factors. The principle underlying PTLD is a consequence of immune suppression given before transplantation and ongoing immune suppression after transplantation. This immune suppression suppresses T and B cell function significantly. If a person were to develop a primary infection or reactivation of EBV during this vulnerable time, the virus could replicate unmitigated, causing B cell immortalization and ultimately tumor formation. One strategy that many physicians employ in managing this problem is reducing immune suppression, hoping that host immune response may control this infection. On occasions, the transplanted organ is sacrificed to allow for the complete stopping of immune suppression and gaining control of EBV. In these well-characterized disease states, the pathways for immune suppression are very well known and treatable, as the outcomes are very common among these outlined pools. In normal populations, however, there are many other ways in which EBV may affect the body, potentially taking an effect ranging in severity based on a variety of factors. One example of EBV's involvement in oncogenesis in normal populations is in the case of classic Hodgkin lymphoma, or CHL. As mentioned previously, by the time one reaches adulthood, it is almost certain that they carry the Epstein-Barr virus in its latent form. However, new research has found that in pediatric and adolescent patients, early exposure may have positive impacts on the treatment of classic Hodgkin lymphoma. EBV-positive status seemed to improve treatment efficacy, increase the likelihood of survival, and prolong survival times in cases of certain death. It was found that in pediatric patients, the tumor microenvironments were found to be significantly more cytotoxic than those of adult patients. Pediatric tumor microenvironments are generally Th1 dominant and therefore possess the CD8 cells necessary for killing virus-infected cells, meaning the cells of the tumor. In children, the tumor is surrounded by a minimally immunosuppressive environment and is therefore highly responsive to treatments, as the body is not working against itself when responding to chemotherapy. The tumor microenvironments of adult tumors, however, are far more Th2 dominant, meaning they possess CD4 or support cells that do not place their focus on attacking the infected cells. These environments also contain a significant amount of Tregs with the FOXP3+ protein, which in normal cases would limit a dangerously overactive immune response. However, by limiting the immune response here, it creates the exact suppressive environment, which is not conducive to accepting and effectively receiving treatment. The tumor environment in pediatric patients is constructed very effectively for responding to direct treatments, creating a positive case when someone has EBV status. However, in adults, the opposite is true, where a positive status means increased immune suppression and reduced immune response. Not all pediatric cases have had good effects. Some have just been neutral. However, an overall observation in adults has been that the exacerbation of classic Hodgkin lymphoma exists, especially because of this tumor microenvironment. These conclusions, of course, can be drawn to an extent. However, it is still unknown if EBV is causing the specific tumor environment or if the specific tumor environment is the one impacting EBV's role. Further, in other studies of the broader population, specifically on cohorts of patients with nasopharyngeal carcinoma, a link between EBV expression and early signs of cancer development was revealed. Nasopharyngeal carcinoma, or NPC, is a cancer of the head and neck that starts in the tissues of the nasopharynx. In an experiment involving serological testing of antibodies from patients who had nasopharyngeal cancer, it was revealed that those in a risk pool for NPC were highly likely to develop a tumor if they had IgA antibodies that developed against EBV's EBNA1 antigen. This correlation is significant in advancing scientific knowledge, as IgA against EBNA1 has now been identified as a biomarker for nasopharyngeal carcinoma, and high-risk groups can now undergo regular blood testing to determine if they will eventually develop NPC. This testing allows patients to learn around 4 years in advance whether they will develop a tumor, which is extremely helpful in planning treatments and quickly identifying problems instead of allowing them to get worse unnoticed. Extensive research into such a link, particularly in the case of over 9,500 Taiwanese men, showed that high antibody levels, particularly of the VCA-IgA antigen, preceded the development of NPC quite dramatically. A positive test for significant amounts of this antigen was directly linked to a 22-fold increase in the development of NPC. This is a vital development, as in a recent study observing the importance of EBV blood markers as an indication of NPC, researchers found that NPC alone is a highly treatable disease when detected early. Early detection was found to correlate with a 90% 5-year disease-free survival rate, as opposed to a less than 50% chance of survival with late detection. These tests were conducted in a population with a relatively high degree of genetic predisposition to NPC. Each subject had at least 2 first or second degree relatives who had been affected by NPC. Here, NPC rates were as high as 100 per 100,000 cases, per every 100,000 cases. Some of these at-risk geographical populations are shown in the map on the left. However, there are a variety of factors affecting predisposition for NPC beyond that of just genetic association. In a control study, interviews indicated that higher NPC risk was often associated with a past of ear, nose, or throat diseases, exposure to fumes and smoke, exposure to chemicals, and environmental carcinogens from certain food groups. Research has also found that EBV DNA found in plasma is also another potential biomarker for NPC, which when used in screening had a sensitivity of 96% and a specificity of 93%. The EBV DNA that is found in plasma is in the form of short fragments that are specifically released by the NPC cells. The presence of this DNA is enough to indicate that NPC cells are present, thus making it a potential biomarker for future cancer development. All plasma and serological tests for EBV DNA or anti-EBV IgA antibodies generally have very low positive predictive values, as is often the case with many asymptomatic diseases. However, there is evidence to suggest that these screening tools may be very valuable for those with known predispositions and risk factors, such that they can take preventive action to prevent future tumor development. These tests are also found to be very cost-effective in these at-risk populations, making them a very good option. It is important to note that NPC is a relatively aggressive cancer with rapid metastasis. However, a 3-year rate of progression-free survival in a group of NPC patients identified that those diagnosed during stage 1 had a 97% chance of survival as compared to a much lower 70% if diagnosed in a later stage. NPC patients were 10 times more likely to have a positive outcome with early diagnosis than later diagnosis because there are current treatments for early stage NPC. Late-stage NPC is very difficult to cure, and therefore, screening for EBV may be valuable amongst those who are identified as being predisposed. Though it is difficult to say that there is a strong enough correlation to suggest any form of immunization, there is certainly evidence to suggest that serological testing and plasma testing, along with subsequent screening, can be very beneficial in these specific populations. Now, finally coming to my conclusion, the failure of many previous research bodies has been in identifying how understanding the pathways EBV takes and the circumstances precipitating them have a certain prognostic value. While there is no clear evidence to suggest that EBV is directly related to the development of any of these diseases, there are strong correlations to suggest the value of the relationship. Understanding these repeat indicators can allow us to develop screening tools using serological and plasma testing, specifically for use in at-risk populations. And thus, we can prevent some cancers altogether and improve treatment and survival outcomes in cases of people who already do have them. There are significant gaps when it comes to the prevention and early detection of EBV-related cancers. So despite the more primitive nature of some of the findings discussed in this study regarding specific technologies, these are still the best option to begin addressing some of the health issues as they present themselves increasingly in today's world. Improving existing screening and preventative technologies is critical given the multitudinous instances of EBV-related oncogenesis. Technological advances present a much healthier future as early detection and prevention will drastically improve treatment efficacy in patients and increase long-term survival rates. The important next step forward is to further clinical research for vaccines, screening, and improve preventative efforts, as an improved understanding of the extent to which EBV can result in a cancer may improve the lives of thousands in at-risk groups. Thank you to my mentor, Max Sacker, for the guidance and support throughout the development of this project, and thank you all for taking the time to listen to my presentation today. Thank you.